Okay. Whereas we said the autonomic nervous system is involved especially in involuntary control of smooth muscle, cardiac muscle, and exocrine and endocrine glands, the somatic nervous system is made up of motor neurons which stimulate skeletal muscles, which we can use then voluntarily. So we usually consider these motor neurons lower motor neurons to differentiate them from the motor pathways in the spinal cord, which are sometimes referred to as upper motor neurons. While we talk about the somatic nervous system as being under voluntary control, you should recall that our skeletal muscles have some reflexive activity, which are involuntary. Our breathing we don't typically need to think about, so we do have involuntary control of breathing as well. And we also typically, if you're standing or sitting upright, you're using some amount of tone and balance of muscles, but you're not having to consciously think about it. And so while you are capable of voluntarily stimulating any of your skeletal muscles, we often have contractions that we don't need to think about and that are happening involuntarily. A couple of conditions associated with loss of motor neurons are loss of motor neurons caused by poliovirus, famously in our President Franklin Delano Roosevelt, who is not able to walk after middle age. And in amyotrophic lateral sclerosis, which is also called Lou Gehrig's disease. In this case, it's caused by inappropriate or ineffective autophagy, and the inability to get rid of waste products results in an accumulation in these cells, and then these cells become irreversibly damaged and die off. So the somatic neurons, compared to others, their effector organ is going to be, again, skeletal muscle. Now they can be influenced by EPSPs and IPSPs, but when they stimulate skeletal muscle and release their neurotransmitter, which is going to be acetylcholine, it's only going to have an excitatory effect. And comparing the autonomic and somatic nervous system, both branches of the efferent division, ventral versus lateral horn, we have lumbar and thoracic, cranial and sacral, but here we're going to have all throughout the spinal cord. Here we're going to have a 2-neuron chain, pre- and postganglionic, single neuron, involuntarily controlled glands, skeletal muscle. Now, the effector organs are going to be innervated by motor neurons, whereas we might have both branches in some cases, the autonomic nervous system. So the somatic motor neurons are always going to release acetylcholine, which is going to act via nicotinic receptors on skeletal muscle, as we'll see in a moment. It's going to result in only stimulation of muscle, the type of control, voluntary, but some can be subconsciously coordinated, like respiration and tone and balance. A bunch of higher brain areas influence the somatic nervous system, whereas our higher brain areas associated with autonomic control are a little bit more limited. So it's important to remember that the somatic nervous system, our motor neurons, innervate multiple muscle fibers. So the smallest amount of contraction you can achieve voluntarily is the stimulation of a single motor unit. So a motor unit is a motor neuron and all of the fibers that it innervates. So if you stimulate this, you would stimulate all of the fibers that are a part of that motor unit. And they stimulated a place called the neuromuscular junction. And you can see the neuromuscular junction here. And the largest portion of this is the terminal button, or in French, terminal bouton. And I'd like you at some point to try and draw this figure. We've already gone over this in lecture, covering the events at the neuromuscular junction, which are very important. You can fill in this blank figure. It's on figure 7-5 in your textbook. And so the events at the neuromuscular junction are first action potential propagation down to the terminal button via saltatory conduction. All of our motor neurons are myelinated. where we have voltage-gated calcium channels. The events here, again, very similar to those we observed at a synapse. So opening of voltage-gated calcium channels, calcium enters. Calcium is going to signal release of vesicles of neurotransmitter, but in this case, the neurotransmitter is a specific neurotransmitter, acetylcholine. Acetylcholine will traverse this synaptic cleft and bind with nicotinic cholinergic receptors on the sarcolemma, so the muscle cell membrane, the sarcolemma. And this region of the muscle cell membrane is called the motor end plate. And in fact, if you were wanting to stain for the motor end plate, you would stain either for nicotinic acetylcholine receptors or the enzyme acetylcholinesterase, which we'll cover in a minute. So acetylcholine will bind with nicotinic cholinergic receptors, and that will allow a lot of sodium to enter because it's going to be going down both its concentration and electrical gradient, and a little bit of potassium to leave down its concentration gradient. Its electrical gradient favors its staying put. This is going to cause a graded potential in the end plate region called an end plate potential. And this end plate potential is similar to an EPSP in that it's going to cause depolarization, but it's different than an EPSP in that it's usually several times stronger, 3 to 4 times stronger than is required to get an action potential to spread down the sarcolemma. So if you get an end plate potential, you are always going to get an action potential. Whereas at other synapses, if you had an EPSP, just a single EPSP, it's unlikely to bring the membrane of the postsynaptic cell to threshold, and thus you would not get an action potential. So end plate potential here is going to cause an action potential to form down the sarcolemma, and then eventually to terminate the signal, we have to have some way of getting rid of the acetylcholine. And we have an enzyme, especially in high concentration in the secondary synaptic clefts, these invaginations of the muscle cell membrane, where acetylcholine is broken down by acetylcholinesterase. So those are the events at the neuromuscular junction. And it looks usually a little more like this. You can see here the terminal button. You can see all of these vesicles of acetylcholine. You can see some mitochondria because there's a lot of energy requiring process. Here is the primary synaptic cleft. Here are secondary synaptic clefts. And you can see here then all of this is the sarcolemma, and these are a bunch of myofibrils within the muscle fiber. And you can see a number of mitochondria here clustering underneath the membrane. It's important to remember that these structures rarely look exactly like these cartoons in your textbook. One of the first places where acetylcholine and nicotinic receptors were first isolated from were in the electric organs, the electric eel, where they have a large amount of nicotinic receptors allowing for rapidly being able to turn on electrical activity through their electric organs. Now, there are a number of diseases and conditions that are associated with influencing these activities. Just a reminder, we mentioned some snare proteins, V snares and T snares, and there are a variety of snare proteins that are required for the acetylcholine vesicles to fuse with the neurolemma, the nerve cell membrane, and thus allow for exocytosis. I'm not going to ask you to name each of these different types of snare proteins that are found there, but some of these are differentially affected by different toxins and drugs. A couple of these problems in the neuromuscular junction are described in page 244 to 248 of your textbook. First, black widow spider venom. Black widow spider venom causes explosive release of vesicles of neurotransmitter of acetylcholine, so you'd release all of these vesicles. This would cause the subjects to contract all of their muscles, and it causes what we call a spastic paralysis, where the subject is paralyzed because all the muscles are contracting and can't relax. Eventually, acetylcholinesterase would break down that acetylcholine, but there are no more acetylcholine vesicles to be released, and so it might become impossible then to stimulate the muscle voluntarily. Black widow spider venom actually includes a couple of different types of toxins. That's one type of neurotoxin it includes. The botulinum toxin is one of the most potent naturally occurring toxins known to man. If you were to take a 200 milligram Advil or ibuprofen tablet and divide it into 200,000 parts, that amount could be fatal for an adult human. It is caused by the bacteria Clostridia botulinum, which is an anaerobic bacteria. And the Clostridia botulinum toxin was going to prevent normal snare protein function. And as a result, the vesicle can't fuse with the membrane and release acetylcholine via exocytosis. So the botulinum toxin, instead of causing a spastic paralysis, will cause a flaccid paralysis, where the muscles can't be stimulated and thus remain relaxed. Via whatever type of paralysis you achieve, one of the biggest problems is respiration, which is dependent upon skeletal muscle contraction. So it's possible individuals may temporarily need to be placed on a respirator. Curare is a drug, a toxin from plants that's used by some indigenous tribes in South America to hunt with. It is a nicotinic receptor antagonist, so it binds with nicotinic receptors and prevents acetylcholine from doing so, in which case these channels can't be opened and you cannot cause an end plate potential and action potentials, and it results in a flaccid paralysis. The drug Panocuronium, it has similar effects. It's also a nicotinic receptor antagonist, and it's used sometimes during surgeries to prevent the subject from moving. It occurs to me, I forgot to mention that botulinum toxin is used as Botox, where very small quantities are injected to highly localized regions to prevent muscle contraction in the face to get rid of wrinkles. Myasthenia gravis is an autoimmune disease where the subject produces antibodies that partially inactivate a portion of the nicotinic receptors, which results in muscle weakness and potentially fatigue. And organophosphates are nerve agents. They've been used as nerve gases, and they're also used in some pesticides. So what they do is they inhibit acetylcholinesterase and thus prevent acetylcholine from being broken down. And that results in a chronically elevated acetylcholine and a spastic paralysis where the muscles are all contracting.